So ChIP-seq and ChIP-seq was very early days performed on yeast. At that time, you probably remember yeast is about 50% coding. The gene is like 1 to 2 kb long, and then the sequence in between the genes are usually also about 1 to 2 kb long. And so very often, the sequence right in front of the gene regulates this gene. And so you do ChIP-seq, you get a target. It's very easy, right? If it binds in front of the gene, yes, it's going to regulate that gene. But then once you look at the transcription factor binding in human or mouse, this is what you normally see. So a core promoter is only 1%. This is like 1 kb from the transcription start site. In yeast, so we only look at 1 kb. In human, if you look at 1 kb, out of all those binding sites, that's only 1%. If you look at maybe 3 kb, you will probably get another 3% binding site. And there are some, so 48 is in introns in the gene. And also, if you look a little further in front of the gene, intergenic is just between genes. There are big gaps between gene sequences, right? If you look at things that are less than 25 kb away, that's another 8%, but there are also a lot of intergenic sequences that are over 25 kb away from any nearby genes. And so there are a lot of these in the middle of nowhere introns or intergenic sequences that where most of the transcription factor binds. So right now, it's still really an open question. How do you really assign the target of a gene given that this transcription factor binds to 10,000 places in the genome, right? So if you say, okay, I would say in the early days when people just did the ChIP-seq or ChIP-seq, they say, okay, anything that has a binding in the promoter, you'll get some result. You get, a few hundred genes that has a binding right at the promoter. You also have to ask yourself, well, for promoter, do you mean 1 kb, 2 kb, 3 kb in front of the gene or a little bit inside the gene? Because it's from transcription start site, it's before and after, right? A little bit. How do you define this? It's kind of an open question. Other people also try to assign a peak to the nearest gene. So for example, if I have a peak and that gene is 10 kb and this gene is 5 kb, it's going to be assigned to the 5 kb and not the 10 kb, right? So people also do that. The distance to the nearest gene transcription start site. Later on, people also notice that the number of binding sites near the gene for a transcription factor is also important. You can imagine if you have one gene and nearby you have just one, say, estrogen receptor binding site. Another gene you look at nearby, there are like 10 binding sites. Usually we would believe that the one with 10 binding sites are more tightly regulated by the TF of interest. Whereas if just with one, we're not even sure whether it's working or not. And if you want to use other knowledge, you can still do that. This is still an open question. And I can tell you some general approach that my lab use, and I think it would work most of the time. I mean, it's, I think, as good as any other method out there. So binding, it will tell you which transcription factor is likely, which genes are near this transcription factor binding. Let's see. So if you have a binding site, whether it's right next to the gene versus really far away, there's a difference. If it binds right close to the gene, it's more likely to have an effect. When it binds so farther away, it's not as important. So far, what we have found that is for most of the transcription factors, their binding usually is the distance from transcription start site. Whether it's upstream or downstream is mostly symmetric, the effect. But then, whether or not that binding will really have an effect depends on whether its collaborators are available. So supposedly this guy likes to play poker, but he's only by himself without the other 3 friends, well, then it's harder to play, right? So the collaborating transcription factors are important. And we also need to look at the gene expression. If you compare the situation when the TF is active versus inactive, and you look at the genes that are differentially expressed, that also gives you an idea that this TF is important. And so in terms of modeling the binding effect, we will have a later lecture directly on chromatin interaction looping. But just to give you a general idea, people notice this potential exponential decay, which means that if the binding is very close to the gene, any gene, then that gene is more likely to be regulated by the transcription factor. But if this is for interactions, but if something is really far away, they are less likely to even loop into the promoter or the transcription starter of the gene, then they are less likely to regulate. And so this type of chromatin looping is important. And the looping generally have this exponential decay characteristic. And with that, based on the binding, we try to assign basically what we call a regulatory potential, which is the sum of binding sites weighted by the distance from transcription start site. So for example, if I want to know what is the regulatory potential of this transcription factor, if I know it's ChIP-seq in terms of regulating this one gene that I'm interested in, I can look at this, so we can have this exponential decay idea. So here, we look at the transcription start site of this gene, and then every time we see a binding site, we ask, how far is that binding site from the transcription start site? And this is weighted by this exponential decay, which means, for example, if the decay distance is 10, actually for a lot of transcription factors, we use a decay distance of 10, which means that if a transcription factor binds right on the transcription start site, that counts as one binding site. But if it binds 10 kb away, it will only be half. And then if for every distance, it decays even more, it's exponential. And so you can sum all the binding sites weighted by their distance with this exponential decay to say, based on the binding, how likely is this gene a target of the transcription factor you are interested in? Okay, so that's just on binding. And that will give you a rank ordered list. This list you can directly download from Systrom. Based on just binding, we will tell you, what are the likely targets of this gene from the top to the bottom. And this is usually, if you see a 2.1, 1.4, 1.4 would probably mean one binding near transcription start site, another one maybe a 10 kb away, something like that, right? And the second one is to look at the gene expression. The gene expression can be based on two different things. Supposedly, so we developed this algorithm a long time ago. If you have a lot of people come to our group to say, oh, I have two cell conditions, one with the transcription factor active and one with the transcription factor inactive. And then we also did the ChIP-seq when the transcription factor is active. We know where it binds. Can you tell me what it does? And so based on the binding, we can calculate what are the putative target, but also we need to look at the expression. We say, okay, when the TF is active or inactive, what are the differential genes, right? So you know which genes are upregulated, which genes are downregulated. You can also look at the gene expression correlation in the tissues or tumors if you don't have differential expression. You just ask what other genes are correlated with your transcription factor of interest. And that will also give you a rank ordered list. And based on those, at the end, we say the rank 1 times rank 2 will give you the final rank ordered list of the direct targets of your interest. And by the way, in terms of transcription factor regulation of gene, it's not a black and white. Every transcription factor has an effect on every other gene. It's just more effect or less effect. It's a continuous value. And so at the end, this rank order is also continuous. You say the top ones are the potential direct target. The remaining ones are weaker and weaker and weaker. And you can take the top ones. So in fact, most of the, I would say, minority of the binding sites is functional. You might see 10,000 binding sites, but it only regulates probably a few hundred genes. And at least the strong ones, the important binding sites are probably in the order of a few hundred to a few thousand, a couple thousand. The remaining ones are just kind of weak. And once you take the top target, do the rank product of the two, one from the binding, the other from the gene expression change. You get the top ranking genes. You can do gene set enrichment or gene ontology. You ask, okay, what is this transcription factor's direct targets do in that cell? And then, by the way, this is now all done with one web server you can use, which will be in homework 4. Okay, so that will give you the target. So yeah, in summary, basically ChIP-seq, you can use it to identify genome-wide all the in vivo in the cell, what the transcription factor is doing in terms of binding in the genome. You can do use max for ChIP-seq peak calling, and there are many, many different QC parameters. There are conservation, FRIP score, FDR, fold change, to help you figure out and also look for motifs. But then once you have the ChIP-seq data, you want to look at transcription factor motifs. And from there, you might be able to find that it's collaborating with something. And also, you want to look at its target. By the way, sometimes a transcription factor can have effect both upregulating gene and downregulating other genes. And that is because they are hanging out with different friends. With this group of friends, they will do good things. With this group of friends, they'll do bad things, or they'll have different functions. And so that can happen as well. But once you figure out the target genes and the collaborating factors, you can also look at the gene ontology of the target genes and then do pathway analysis. Okay? Yes?
Can you explain again how can we rank genes based on binding?
So let's look at this situation. We look at every gene and look at all the binding sites within 100 kb from this gene, right? And if a binding site is right as transcription start site is weighted 1, it's binding further away, then it's weighted less and less and less. And at the end, we add up everything weighted by the distance to get a score. And so what kind of gene will get a high score? They have more binding sites, and those binding sites are close to the transcription start site of that gene. They will get a good score. They will be the higher ranked target. Whereas the gene has either no binding sites or there's some weak binding sites that are really far away, then they will have a very low score. Okay. One thing I forgot to mention that the decay rate, what is the decay? What we found is that there are general 2 types of transcription factors. One type only have very short range effect. They have effect usually within only 1 to 2 kb away from the binding site. And you can see that as well. Their binding have probably 20 to 30% really bind at the promoter. It's still a minority, but it's like 20 to 30. Or even 10% is kind of rare. Whereas some factors have really wide range remote control type of transcription factor that have 10 kb or sometimes even longer. Their binding site, you look at genome-wide, there would be like 2% binding to the promoters. So just looking at the binding distribution, at least our group can give you a rough estimate of what that decay distance should look like. Okay, but these tools are all available now. You can use. Okay, great. That's good for today.